Let's go ahead and do our notes today on DNA replication. So we'll be going through slide 10 through 17. So what is exactly replication? It's the copying of the DNA molecule. We talked about this in mitosis, meiosis, that type of thing. We have our chromosomes, they're replicating or they're duplicating. That's another word for it. What happens during replication is first we have an enzyme and it breaks down the hydrogen bonds between the nitrogenous bases. Now remember, those nitrogenous bases are the bases that are in the middle. They make up the rungs of the ladder and they are put together by hydrogen bonds. And so those unzip from the middle, pulling each side of the ladder apart from each other. This frees nucleotides. Then we have free nucleotides that are out in your cytoplasm are going to be coming back in and they're going to be attaching to the bases along each strand. So as we unzip, each part that is a single strand now becomes a template for a new double strand of DNA. And nucleotides come to match back up, A with T and T with A, C with G, G with C. Then another enzyme is going to bond those nucleotides into a chain. So essentially what I've done is I've unzippered my DNA, put new nucleotides on each template that was made, and now I have two new copies of my DNA, identical to what I started off with. Let's say this is one side of a DNA molecule, all right? GGTAAC. Then our second side would be C, because G and C always comes together. G is going to go with C. T is going to match up with the A with T, A with T, C with G. That would be my second DNA side. Now, look at this picture because it's kind of interesting. I want you to understand it's not like we take the whole great big DNA molecule and unzip it all the way to the end, because remember how long this is. It's what happens is it unzips, gets put back together, unzips, gets put back together, and then I end up with my two as I go along, as it's working its way up. It's unzipping and making its new copy. If you can tell, it has been unzipped from the bottom. We have our newly formed, and this is my region in the middle where I have my two templates. Where it says region of replication, understand that each side there is a template, and then we go ahead and put the DNA, the nucleotides that go, the nitrogenous bases that go there in there. So from DNA, we also are going to get our proteins. Oops. All right, so from DNA, we're going to get proteins. And so how does the information found in DNA molecules code for an organism? Well, it codes for it because the sequences of those nucleotides is set in instructions for making proteins. And when those proteins are made, then it then gives our instruction. To make proteins, though, we need to have ribonucleic acid, RNA. Now notice that it's very similar in terms of its name as DNA. The difference is R, and we'll explain that in a minute, versus the D. It is composed of a single strand of nucleotides. So remember, RNA is a single strand, not a double strand. It is made of three parts again. Simple sugar, which is our ribose here. That's where the R comes from, not deoxyribose, but ribose. That's where the R comes from. Again, it is made up of a phosphate group, and it is made up of a nitrogenous base. Just like DNA, the difference is the simple sugar. The bases here are different, though. It can either be adenine and guanine, or cytosine and uracil. So uracil replaces the thymine. So the thymine and the uracil and the cytosine are similarly shaped. So if you go back in our notes and you look at those different shapes of those nitrogenous bases, uracil and cytosine are similarly shaped. Uracil is in RNA where thymine would be in DNA. So what happens is, if you notice, the RNA on the left-hand side is a single strand, unlike your deoxyribose nucleic acid. Notice that the difference between the thymine and the uracil here, those are the two things that are different. So A and U are going to come together, C and G, whereas in the other one, we have the CG and AT. All right? So we have our singles along the edges, and they could be adenine, uracil, cytosine, or guanine. That's what that AUCG means at the top. The ATCG means it could be adenine, thymine, cytosine, or guanine for your DNA. All right? So RNA molecules are used to build proteins from the set of information provided by the DNA. So the information on how to build the RNA comes from our DNA. There are 3 types of RNA that we're going to talk about, and you need to know what these 3 types are. The first type is messenger RNA, which is little m RNA. And what this does is it brings the instruction from the DNA and the nucleus to the cytoplasm. So it is what contains the instruction. DNA tells us how to make it, and the carrier of that instruction is the mRNA. So it takes that information from the nucleus out to the cytoplasm, and that's important for you to know where it starts and where it goes to. The second type is ribosomal RNA, which is rRNA, and it is what makes up the ribosome. Remember from our chapter where we talked about the organelles, the ribosome is the site of protein synthesis. So this is where our proteins are made. Ribosomal RNA makes up those ribosomes. The third type is what we call transfer RNA, or tRNA. This one does exactly what it sounds like. It transfers the amino acids to the ribosomes so it can be made into a protein. So it delivers the amino acids to the ribosomes so that it can be assembled into a protein. Now, we have our messenger RNA, which has the instruction on it. It sends the message on what to make. Ribosomal RNA, which makes up the ribosomes, which is where the proteins are made. And the transfer RNA is what gets the items needed to the ribosomes to actually make the proteins. So if you kind of look at this blob right here, you can have your UCCAAUG down at the bottom. And what happens is that we will get the complementary pair for these, and those complementary pairs will then dictate what the amino acid is that is going to be put there. And then your line of amino acids is going to dictate what the protein is that is being made. And we'll talk about that more when we talk about protein synthesis. But before you can talk about protein synthesis, you do need to know what the 3 types of RNA are. And remember, those are messenger, transfer, and ribosomal. You're going to need to know what all those are and what they do so that when you get your checkpoint today from your notes, that you know what all those are. All right, so that is the second portion there of the notes for replication and RNA.